Everyone. My name is Elizabeth Stulberg. I represent the Office of Science and Technology Policy. I'm the Senior Advisor for Food and Life Sciences. I'm also an Agriculture Science Fellow at the USDA's Office of the Chief Scientist. And I do know that it is June. Sorry. I'm sure everyone has that experience when you look at your slide and you think, no, that did not just happen. So thank you very much for being here. I was asked to come and talk to you about the National Microbiome Initiative, which is an initiative run out of the White House, and to try to give what you're doing in the built environment some context within the national efforts for all microbiomes. I don't need to tell you probably that microbes are everywhere. This is a group, I think, uniquely that understands that. They're in especially unexpected places, and they are consequential in those unexpected places. They can cause disease. They can prevent disease. Microbes can pollute, but they can also clean up. And there are some microorganisms that can do all of these things, depending on the context. So as a plant biologist, a plant molecular biologist working in soil microbe interactions, it was my pleasure to be able to work with Dr. Jo Handelsman, who is the director of the Science Division in OSTP, the Office of Science and Technology Policy. Jo is a microbiologist. She actually coined the term metagenomics, so it was very exciting for me to get to work with her. And she introduced me to the fact that not only are microbiomes important, which I already knew, but they're actually integral to many presidential priorities, policies and ideas going on at the White House already that we can plug into. Things like precision medicine, energy, agriculture, and climate change. Understanding microbiomes and working with them are going to be essential to working through these challenges. So Jo had a vision. She had a vision of a national microbiome initiative, and that was clear from the time I started working with her. And we put our heads together to try to figure out what exactly this national microbiome initiative would look like. And we had to start by saying, well, what's already being done? Because that was not a very well thought out thing at that time. And what needs to be done? What's next? And what specifically can the White House do? Nobody wants government overreach into your science, right? So we just need to make sure that whatever we're doing is going to be welcome to the scientific community and the community of microbiome people at large. So let's start out with what are we already doing? Keeping in mind that the federal government is large and it's doing or funding a lot of science. And there's no massive scientific database. There's no giant list of all of the research programs where you can just do a Control F microbiome. It's not there. It's not. And so to even parse out the difference between microbiome research and microbiology research was a challenge. Where do you draw that line? So what we did was we chartered a committee called the Fast Track Action Committee for Mapping the Microbiome. It convened members from 12 different federal departments and agencies. We developed a survey that measured both quantitative and qualitative metrics of microbiome science. So this is things like quantitative things like how many projects, how much money, which ecosystems, but also qualitative things. We asked the scientists and we asked the program managers, where do you see this research going next year, 5 years from now, 10 years from now? What are your needs, technological needs, tools? What are your human resource needs? And what are the big challenges or roadblocks that are preventing this field from getting to where you think it needs to go? We asked all of these questions. And we published a report 90 days later. 90 days later, we published a report, and it included responses from all of these departments and agencies. And I list them just to say, there are a lot of scientists working on microbiome in places that you wouldn't necessarily expect. Well, maybe you would, in this room, you might expect, but that I didn't necessarily expect. And actually, once we started this process, we had more and more interest. The VA came to us and said, wait, we need to know about this too. USAID, Agency for International Development, they came on board as well, very enthusiastically. We want to help smallholder farms, and microbiome in agriculture is a really important thing, they said. So we really expanded our scope. We ended up doing the survey with 14 different departments and agencies, but I think there are still pieces that we missed. All right, so while all this is going on, in this 90 days, we're developing the survey, we're giving the survey, we're collecting the responses, we're writing the report. We also convened a bunch of scientists at OSTP. So these are scientists that aren't in the federal government, but we asked them the same questions, the same types of qualitative questions. What do we need to do? Where is this research going? And to cover as many bases as possible, we issued a request for information in the Federal Register. And for, the few of you in the room who don't read the Federal Register every day, we also published a blog on our website, which I'm sure you do read every day. And we said, please, tell us what you think. And we got so many responses. It was actually really fun to read through them. Well over 100 responses, some of them from scientists, some of them from people in industry, some of them from your average person. Some of them were heartbreaking. My wife has a C. diff infection. Can you please hurry up? Can you please hurry up with this fecal microbiota transfer research? Because something has to be done. And it really brought it to a personal level for me as well. And I'm sure many of you who have clinical experience in the room feel the same way when you talk to people and you say, no, we really have to understand this. We have to get this under control. So what was really striking to us about all of these endeavors, the report through the federal committees, the convening of scientists, and the public request for information, were the themes that emerged that cut across ecosystems. It didn't matter whether you were working in medicine or in marine biology. What we saw was, these are the questions. What is a healthy microbiome, and what is the nature of robustness? In other words, what makes a microbiome resistant to change or resilient? And the tools and technologies that people said they needed were also very similar. We need tools to detect, measure, and model in multiple dimensions the chemical currency of microbial communities, how they communicate with each other. We need to develop computational tools because we have these vast data sets coming in that we need to analyze. And we need to be able to model community behavior. We need that software. And all of this is in order to alter microbiomes predictably and reliably, because what good does it do you to know what a healthy microbiome is if you don't have one? And it doesn't matter whether you're a doctor or an architect or anything else. You need to know how to change the microbiome to something that you want to see. So what could the White House do? What could we at OSDP do? Well, we can promote investments in microbiome research, and we can facilitate interdisciplinary research. We can make that just a little bit easier, because we think it's important for medical scientists to be able to talk to plant and soil scientists, and to be able to work with marine biologists. And all of them really should be working with microbial ecologists and data scientists and engineers. This is a very interdisciplinary sport, microbiome science. And what was surprising to us, cat's out of the bag, by the way, in terms of the federal investment in research, we studied from fiscal year 2012 to 2014. We found that the U.S. government was investing more than $900 million over those 3 years. But it was very siloed. It was this is what USDA is doing, this is what NIH is doing. And they didn't necessarily talk to each other because, again, they didn't necessarily know that the research they were doing was microbiome research. It wasn't even really called that. It wasn't listed that in their project title. So they didn't know even who to talk to about it. So what can we do moving forward? Well, the National Microbiome Initiative raised $120 million of US government funds to support more microbiome research and to make it easier to make that research interdisciplinary. We set up a new committee that would facilitate interdisciplinary, interagency communication about this type of research. We also put together $400 million in new commitments from non-governmental groups to support microbiome research, both interdisciplinary and specific. And the goals of the initiative, what we were hoping to achieve, first of all, interdisciplinary research on these fundamental questions about diverse microbiomes. Because doesn't it make sense that if you're all working on the same questions, maybe working together, you can make more headway faster than in your own silos? And then the platform technologies for probing and changing microbiomes. And of course, access to all the data from the different ecosystems. And then, of course, we need this new workforce, people to actually do all of this work. We can do that through citizen science, public engagement, and educational opportunities. To harness microbiomes, the power of microbiomes, and to make them healthy, we need to be able to alter them predictably and reliably. And so we need to understand robustness. We understand how microbes can change or not change. And this is not a new thing. I stole this slide from Jo Handelsman because I liked it so much. This is Ilya Mechnikov. He's a microbiologist from last century, even a century and a half ago almost. And he loved microbes in a time when it was not fashionable to love microbes. In a time when people thought that microbes just cause disease, he consumed liters of yogurt with lactobacillus every day in order to change his own gut microbiome, because he believed that this lactobacillus would prolong his life, would make him healthier. But what he noticed was, as soon as he stopped drinking the yogurt, the lactobacillus would disappear from his microbiome. And I'm sure that was very frustrating for him, but this kind of underscores the point that even his own microbiome was resistant to change when he was trying so hard to change it, and it didn't. And so now I'm going to bring this over to a built environment example. There was an outbreak of listeriosis in ice cream from 2010 to 2015. It was a multi-state outbreak. It caused 10 illnesses and 3 deaths. The Bluebell Creamery in Texas did the right thing. They knew that their ice cream was causing these diseases. They knew that the listeria had been contaminating their product, and they issued a full inventory recall. It was the right thing to do, but it meant that they had to lay off over 1,400 employees and furlough another 1,400. And while this was going on, while these employees were furloughed, they went into their factories and they cleaned them out. And they scrubbed and they cleaned, and they actually had to replace some of their equipment due to this listeria contamination. In other words, they could not scrub the listeria off. They could not be assured that it was clean enough to use again. A lot of the built microbiome research that's been going on, and of course I know mostly about the federal research that's been going on, has been, I think it was only about 2% of the total. And it was mainly NASA, and they were mainly working on trying to keep the space station sterile. It's not. I don't think there's anything. I'm beginning to think that there's nothing left that is sterile in this world, and maybe that shouldn't be the goal. So in this case, they're trying to keep these pieces of equipment sterile. Maybe that shouldn't be the goal. Maybe if we could figure out how to create a microbiome environment on these equipment pieces that can be resistant to change, that won't allow contamination in the first place. Or maybe we have a microbiome that could be an indicator. We know when something changes and could be susceptible to an invading pathogen. Maybe that could have prevented these deaths, could have prevented this economic disaster from this local community. I stole these pictures from the front of the National Academies agenda. I think they're really great. But I just wanted to point out that with the exception of the space station, which is supposed to be sterile, remember, and it has the one person in it, the other pictures, something was kind of interestingly weird to me about them. These are rooms that are not being used for what they are supposed to be used for. They're empty. And it is important for us to understand what's going on in a surface in a hospital, in an operating room, or on a plate, or a tablecloth, or on a desk and a chair where people work. But I implore you, as you're thinking about the microbiome and all of the interdisciplinarity of especially the built environment, to not forget that people use this stuff too, and that people are an extra layer of complexity that we have to consider, and that I think many of you in the room are already thinking about. So in terms of the scope of the National Microbiome Initiative, I think the goals for investigating the built microbiome are very similar. Interdisciplinary research, platform technologies, getting the right workforce involved, absolutely the same. But how people create and interact with their environments and their microbiomes is something that you also have to consider as well. So thank you very much. I'm very hopeful to see the outcome of your work. I'm not sure if I'm supposed to be taking questions.
Burning questions now, and then we can do a longer discussion at the end of the session. Burning questions. One question.
Burning questions. I can stay up here too. Yeah, okay. Yeah.
Can you tell me what might have been the cause and what was the solution?
I'm pretty sure in this case, the listeria came from one of their, not toppings, but like the mix-ins. It might have come in, and then and please, I'm not 100% sure that this is correct, but I'm pretty sure that it came in through one of the machines that, I'm not an ice cream specialist, that loads the mix-ins into the ice cream, and that the listeria established itself in this equipment that puts the mix-ins in, and then for subsequent use, I mean, they cleaned it in between, but they didn't get the listeria off. So then every time they put in, you know, your Oreos or your peanut butter chips or whatever it was that went into the ice cream, it ends up getting into the next batch. Does that answer your question? Is that what you were looking for?
Well, I just can't figure out what the source might have been.
I think the original source was an external food source. That's right. Mm-hmm.